Swayam Prabha. Digital India. Educated India.
Namaste. Today we will go through the second leg of revision session. So, we looked at LIDAR, which stands for light detection and ranging. So, it is we looked when we discussed it, we found out that it is an active remote sensing technique that is also known as airborne laser scanning or ALS. So, it is a combination of laser plus radar. So, it uses a laser light in the same way that a radar uses radio signals. We used laser because it is monochromatic and it is directional. We looked at its concept. So, we need to know the exact position of the aircraft using differential GPS and the inertial measurement units, and then we get distance to the surface as we did in the case of a rangefinder. So, D is equal to C into T by 2, and by keeping track of angles we can get a 3D scan. So, there are 4 components of a LIDAR. We have laser, scanner and optics, photo detector and receiver electronics, and the position and navigational systems. So, these can be used on a satellite, or these can be used on a helicopter, or these can be used on the ground. Then we looked at different scanning mechanisms. So, we can have an oscillating mirror, a rotating polygon, or a nutating mirror, which will give us different kinds of ground patterns. There are 2 families of LIDAR. So, they go by the name of waveform LIDAR and discrete LIDAR. We also looked at the wavelengths that are deployed, that can also lead to a classification of LIDAR into topographic LIDAR that uses a near infrared laser, or a bathymetric LIDAR that uses water penetrating green light to measure the sea floor and the riverbed elevations. So, what is the use of LIDAR in forestry? So, in the case of forestry, we need to know information about the canopy cover. So, in the case of LIDAR, we can get this digital elevation model of the land, we can get the canopies. So, we can even get canopy structure. So, in the form of cross sections at different points or different heights. We can get the height of the tree, we can get the leaf area density using LIDAR, we can digitally calculate the canopy height of all different trees. We can compute all these information together to get carbon stocks. So, this figure tells us how much kg of carbon is there in each of these trees. We can also use a LIDAR horizontally. So, we can also get carbon stocks in horizontally. We can use it to study the plant growth and shape change, and also the growth of forest. So, whether it is a young crop, a mature crop, an old crop, or a mixed of these. Next, we looked at canopy attributes. So, in the case of canopy, they are very important because the size of a canopy will give you the amount of photosynthesis that is going on in a tree, and it also is important in the case of nutrient cycling and so on. So, species diversity, nutrient availability, biodiversity. So, for instance, orchids and are epiphytic plants that grow on top of trees. So, they require canopy. There are some arthropods, there are some mammals such as the sloth. So, sloth only lives in the canopies. And then canopy also is important for the retention of carbon. So, we looked at canopy parameters like canopy diameter and the canopy length or the crown length. This can be used to calculate the canopy volume and biomass. Then we also looked at the canopy cover and canopy closure. So, we can define canopy layers. We can find out the structure of the canopy. So, this can tell us whether the forest is healthy or whether it is a sparse forest or it is a heavy defoliated forest, and we can even use LIDAR to study the canopy. Now, in the case of canopy, we also looked at how do we look at a canopy. So, we stand half to 1 tree length away, and then if there are 2 observers, then they should stand at 90 degrees to each other. We looked at vigor class. So, what do we mean by vigor class? It is a visual assessment of the crown vigor of saplings. With this, we defined 3 kinds of vigor classes. So, class 1 was the most vigor class. So, in this case, saplings must have an uncompacted live crown ratio of at least 35% and less than 5% dieback, and 80% or more of the trees present must be undamaged. So, in this case the saplings will have a vigorous growth. Then we also defined a poor vigor, in which less than 20% of the trees of the leaves are undamaged, that is more than 80% of the leaves are damaged. That does not fall in the first or the third vigor class, is called a moderate vigor class or class 2. Then we also looked at the uncompacted crown ratio. So, to find out the uncompacted crown ratio, we first established the live crown top, then we determine the base of the live crown. So, with both of these we can find out the crown length, then we can find out the height of the tree as we have already seen, and then we can calculate the ratio. Then we also looked at crown light exposure. So, that is an estimate of the amount of direct sunlight that reaches the live crown when the sun is directly overhead. So, in this case we defined units of 0 to 5. So, we looked at our canopy in the form of cones and we divided it into 4 sections horizontally and one section that covers the top portion. So, if any of the horizontal sections receives 100% light, only then it is counted. If it is receiving partial light or no light, then it is not counted, and if in the case of this top portion, if any part of it is receiving light, then it will be counted. So, it gives us an estimate of the amount of direct sunlight to which the tree is exposed. So, that will give us the amount of photosynthesis that it will undergo and the amount of competition and the stand structure that our forests have. Next we looked at crown position, which establishes the location of an individual live crown in relation to the surrounding over canopy. So, here we defined 4 codes, that is super story, over story, under story and the open growth crown. So, in this case we first go for the average height, which will give us an over story zone, then we find out 50% of that height, and that can be used to classify our trees into 1, 2 or 3. And in cases when our trees are grown very separate from each other and there is no competition, there is an open canopy, then we give it a code of 4. Next we looked at some other canopy attributes including crown density. So, crown density is the proportion of crown volume that contains biomass including foliage, branches and reproductive structures. So, we saw that we can figure out its crown density by using by first making out the shape of the canopy and then using some cards. So, these are the foliage transparency scale cards or the crown density scale cards. So, it gives us the percentage of crown volume that contains biomass. Next we looked at crown dieback, so which is an early indication of stress, and we recorded in 5% classes from 0 to 100. Now, in this case we drew a crown outline, blocked the affected area and then figured out how much is the dieback. Next we looked at foliage transparency, which gives us the absence of foliage where foliage should normally have occurred, and it is negatively correlated with tree health. So, if your crown does not have enough foliage, so in that case it will appear transparent. So, all the light that is hitting the crown will be able to pass through, and in that case your tree will not be having enough amount of photosynthesis, and so the amount so its health will also be poor. Here also we define it in 5% classes and we use our foliage transparency cards to determine the amount of foliage transparency. Next we also looked at crown diameter. So, in the case of crown diameter we can take 2 measurements, and with those 2 measurements we can keep both those diameters as the crown diameter values. Now, we looked at the importance of crown measures. So, they are an indicator of photosynthesis. Larger crowns will give us higher growth rates. They are useful in predicting responses to silvicultural treatments like thinning and fertilizer application. They are part of growth and yield models, and the size is a surrogate for or it is a proxy for photosynthetically active foliage. So, we also looked at crown width and the surface area and volume determination of a crown, which is done by considering our crown to be having a conical shape. So, these the surface area and the volume are the equations of a cone. Next we looked at canopy cover and closure. So, we defined both of these terms. So, inside a forest the amount of light can vary depending on the amount of canopy that is there. So, canopy will prevent light from reaching the bottom portions of the forest, and we can measure light using a photometric or a radiometric measure, and also by using proxy measures such as canopy cover and closure. So, we looked at a demonstration of how we can use a photoresistor to measure the amount of light that is coming to a point. Then we looked at canopy cover. So, canopy cover is seen from the top. So, it looks at the projection of all the canopies if they are done on the ground. So, what proportion of the area is being covered by these projections will give us the canopy cover, whereas in the case of canopy closure we take this bottom point, and from that point we take a reading from all the directions, and the proportion of the points that are covered by the canopy or that see a canopy there will give us the canopy closure. So, in the case of canopy cover it varies with the height. In the case of canopy cover height is immaterial, but in the case of canopy closure height plays a very important role. So, canopy cover can be assessed by using a tube or by using remote sensing. So, we looked at these tubes. So, essentially they are periscopes that can give us what is seen on the top, or we can use remote sensing to for instance we can use photography or even lidar to find out the areas that do not have a canopy. And in the case of canopy closure we can use a densiometer or hemispherical photography. So, we looked at what a densiometer is, and it has 96 points, and those 96 points can give us an estimate of the amount of canopy closure. So, all of these, so it is divided into 24 grids. All these grids have 4 points, and any point on which you can see a canopy is counted, and then that value is divided by 96 and then converted into a percentage to give us the canopy closure. Then we also saw this hemispherical photography, which uses a fisheye lens or a very extreme wide angle lens. So, here we can see what portion of the view is covered by the canopy, and that can also give us the canopy closure. Next we looked at photogrammetry. So, photogrammetry is a form of remote sensing that utilizes the science and technology of obtaining spatial measurements and other geometrically reliable derived products from photographs. So, the main principle of photogrammetry is that triangulation permits depth perception. So, essentially we are taking multiple images of an object, and then we are using photogrammetry to convert it into a 3-dimensional information. So, photogrammetry is used for metric applications and interpretive applications. So, we can measure some parameters from a photograph, or we can use our photograph to interpret something, meaning what is actually there in the photograph. So, for instance, in this picture if we say that it is showing us canopies of different trees, so that is an interpretive usage of photogrammetry, whereas if we use it to calculate the crown closure, then it will be a metric application. Then we looked at how do we take good photographs. So, we looked at how field of view, focus and exposure determine what sort of photograph we will get. We also looked at metric cameras, in which all the parameters of the camera are very well defined. There are very low less distortions and a constant focal length. We also looked at stereometric cameras, in which we have 2 metric cameras that are attached to the ends of a precisely measured bar to produce a stereo pair of images. Then we looked at variants of photogrammetry. So, these include aerial photogrammetry, terrestrial photogrammetry and industrial and scientific photogrammetry, depending on where are we taking these pictures, whether we are taking it from the air, whether we are taking it from the ground, or whether we are using it in an industrial or scientific fashion. Then we also looked at far range photogrammetry and close range photogrammetry. So, in the case of far range photogrammetry, the focus is at infinity. In the case of close range photogrammetry, the focus is at a finite distance. Then based on the orientation of the camera axis, we define true vertical photogrammetry, near vertical and oblique photogrammetry. Now, in the case of oblique, we again divide it into high oblique and low oblique. In the case of high oblique, we can see the horizon in all the photographs. In the case of low oblique, we cannot see the horizon. So, we looked at differences between vertical and oblique photogrammetry and when do we use what. Then we saw that in the case of aerial photogrammetry especially, the photographs that are taken have some amount of overlap between two pictures, and this amount of overlap is then used for triangulating different distances. So, there are a number of corrections that are required. So, these corrections can these days be done automatically by using a number of different softwares. Then we can take measurements by using this principle of stereo viewing. So, in all these photographs we have an overlap between the ends and an overlap between the sides, and that can be used to do the measurements. Then we also looked at the applications of aerial photography. So, it can be used to construct a number of different maps, large scale or small scale, where then we can use it to study hydrography of a location, and we can also use it for exploration and recce purposes. Then there are some other products that come out of this science of photogrammetry, like digital elevation models or DEMS, orthophotos, thematic GIS data, and other derived products and maps. Now, in the next section we looked at the basal area of a tree and a stand. So, we defined the basal area as the area that is occupied by the cross section of a tree trunk, usually measured at the breast height, and we use basal area to get an estimate of the stand density, the stand volume, the stand growth, and for forest management purposes. So, why do we use basal area and not the number of trees? So, the number of trees or the number density is a very good indicator in case of plantations with very young trees, but as our trees grow further, so their diameter increases and the amount of space and the amount of nutrients that they require also increases. So, in later cases our number density is not that good a parameter as compared to the basal area density. So, next we saw how do we calculate the stand density or the number density. So, we count the number of trees per sample plot divided by the area of the sample plot to get the stand density or the number density. Then we looked at the calculation of the basal area of a tree. So, if you consider it to be having a circular cross section, then you can use pi r square or pi d square by 4, and if you consider it to be having an elliptical cross section, then you can go for pi a times b, where a and b are the half axis. So, next we looked at an example of finding a basal area. Now, in the case of a stand basal area, it is the sum of the basal areas of all the trees that are there in a sample plot divided by the area of that sample plot, and it is represented in meter square per hectare. So, there are three methods of calculating stand basal area. We can do a sum of the tree basal areas, we can use point sampling methods, or we can go for a spacing factor method. So, we first looked at this summation method. So, in which case we find out the basal areas for all the trees that are there in the area, then we add them up to get this total basal area, and then divided by the area of the sample plot to get the basal area of trees per hectare. Next we looked at the other two methods of calculating the stand basal area, and also looked at crop diameter and crop age. So, in the case of spacing factor method, the spacing factor is given by the average distance between two trees divided by the average stem diameter between two trees, and we saw this theoretical construct in which the spacing factor is related to the basal area of the stand. So, if we know any spacing factor, we can figure out what is the basal area, and if we know the basal area, then we can calculate the spacing factor. Now, spacing factor can be used. So, given our average tree diameter, we can use spacing factor to figure out what is the average distance between two trees. So, next we looked at this example in which we were asked to compute the spacing factor and the basal area of the stand from the graph. So, it can also be used to calculate the amount of thinning that is required, and so we looked at these examples. Next we defined crop diameter. So, crop diameter is used to calculate the volume of the crop, and we use crop diameter or DQ, and we define it like this, that the mean basal area of all the trees in the stand. So, this mean basal area is the basal area of n number of trees divided by n. It is equal to pi by 4 into crop diameter square. So, next we saw this example of how to calculate the mean diameter and the crop diameter, and we saw that crop diameter is always greater than mean diameter. So, we also computed how we can prove this. Next we looked at crop age, which is the age of an even aged crop. So, in this case, to find out a crop age, we take the basal area and calculate the crop diameter. So, crop diameter is given by the mean basal area per tree is equal to pi by 4 d square. Then we plot an age versus diameter curve, and we read the age corresponding to the crop diameter, and that is called the crop age. So, that is not the mean age of the crop, that is a different figure that we get out of the crop diameter. So, it is not from the mean diameter, it is the crop diameter. So, that is something that you need to remember. Next we looked at point sampling. So, point sampling is a sampling that goes by the name of PPS. So, it is probability proportional to size sampling. So, in this case, if we maintain a constant critical angle, so we can use that critical angle to get the sum of the basal areas of trees, and we can also use it to get the basal area of the stand by using the equation that the basal area of the stand is equal to n times the basal area factor. Now, this basal area factor is computed from the critical angle. So, there are two types of point sampling. It can be done in a horizontal fashion to get the basal area, or it can be done in a vertical fashion to get the height. So, in the case of horizontal point sampling, the trees are measured at breast height. You can use any instrument, you can use specially constructed instruments or tubes, or you can use even the end of a tape, or you can use even a penny, or you can use this instrument that gives you 3 different basal areas, or you can use the end of your thumb. And whenever you are using these instruments, then any tree that subtends an angle that is greater than the critical angle is counted as a full tally. Any tree that subtends an angle that is less than the critical angle is not counted, and any tree that subtends an angle that is equal to the critical angle is counted as a half tally. Then we also looked at a wedge prism. So, a wedge prism results in a displacement of the tree. So, when we use it to observe trees in a forest, so we can see some trees that are out. So, when they are completely out, we do not tally them. If they are completely in, then we call it a tally. And when we have a borderline tree in which the right edge of this displaced tree is meeting the left edge of the actual tree, then we call it a borderline tree and we call it a half tally. So, there are a number of factors that affect accuracy in the case of PPS sampling. So, for instance, it is difficult in the case of dense stands. We need to correct for slope, we need to correct for leaning trees. We need to ensure that there is no double counting when we are going all around 360 degrees, and then if there are some hidden trees, then we need to go sideways. Now, these are the general rules for point sampling. We choose a BAF such that the number of counts is between 10 to 12, and then the number of samples is also given. So, now the computations from point sampling. So, we can compute the number of trees per hectare by this equation. So, the number of trees per hectare is the basal area factor that is given from the instrument itself, multiplied by sum of the inverse of basal areas of the tally trees. Then we also have we can also calculate the number of trees per hectare in each diameter class by multiplying the number of tally trees with the basal area factor and dividing it by the basal area of the midpoint of the diameter class. Then we can also calculate the stand basal area per hectare, as we have seen before, it is n into BAF. So, next we saw some examples in which we were asked to compute the number of trees per hectare and the number of trees in each diameter class. So, that was given. Next we looked at the advantages and limitations of point sampling. So, there is no need to fix any to lay any fixed area sample plot. So, it saves a lot of time. Plus, in the case of PPS sampling, the high value trees are sampled in greater numbers. So, because high value trees are what we want to know more about. So, these are trees with larger diameters. So, these are those trees that can be extracted very easily from the forest. So, that is another advantage. There is no need to directly measure diameter for basal area per hectare and volume calculations, and it is quick, especially for reconnaissance surveys. So, even if there is some amount of error or if there are some limitations, even then we can use it for reconnaissance surveys to get a quick figure. Now, we also looked at its limitations. We cannot calculate sampling intensity, it is difficult to calculate, plus it requires some skill plus it is difficult in the case of heavy undergrowth in the case of tropical forest, and then because we only have a value between 10 to 12. So, even a small error in tallying can get exaggerated, and then we need to take care of slope, hidden trees and so on. Then we looked at the estimation of number density. So, number density or the stand density is given by number of trees per area. So, there are a number of methods to do it. We can use a sample plot method, in which we lay out a sample plot of area A, count the number of trees and then divide that figure by the area of the sample plot. So, we then looked at some examples. Next, we saw this method in which we can find out this distance between nearest trees, and then we can use the equation as the number of trees per hectare is given by 4 by pi into 10,000 by Li square, and if we have a number of measurements, then we can take the average of 1 by Li square. So, then we looked at some problems in which we use these equations. Then another method is to compute distance not between these nearest neighbors, but between the nth nearest neighbors. So, in that case we say that the number density is n minus half into 10,000 divided by m pi into the sum of 1 over k I square, where k I is the distance between the nth nearest neighbors. So, then we also looked at some examples. Then next we looked at some other sample calculations. So, we looked at these methods again, and then we looked at this crown by bowl diameter. If that is given and if that is fixed, then we can figure out the maximum feasible basal area of a stand per hectare given certain kinds of spacings. So, for instance, if you have a square spacing, then the number of trees per hectare is given by 1 by k square into 10,000, and the basal area of the stand is given by 10,000 pi by 4 z square. So, this crown by bowl diameter is written as z. So, just by using this figure you can calculate the basal area of the stand. Next, we looked at some other examples of how to use the basal area factor. In the next section we looked at volume calculations. So, volume calculations can be done through sections. So, like in this example in which we have cut artery in standard sizes and then we take the measurements. Then next we looked at the volumes of different forms. So, in the case of a cylinder we use pi r square into h, which can also be written as S into L. Then we looked at Smolyan's formula, Huber's formula and Newton's formula for different solids and their frustums. So, Newton's formula is considered to be the most accurate formula. So, in this case we multiply the length of the log by with 1 by 6 of S1 and S2. So, S1 and S2 are the end surface areas or the end basal areas, and 4 times of the middle basal area. So, that is Newton's formula. In the case of Smolyan formula, we take 2 measurements of the 2 ends of the log, then take the average, multiply that with the length. And in the case of Huber's formula, we just take 1 measurement at the middle and we multiply that with the length. And then we saw that in the case of Smolyan, and so Newton's formula is used to calculate the errors in different other formulae. So, in the case of Smolyan's formula and in the case of Huber's formula, the errors are on opposite sides of 0. Now, we also looked at the quarter girth formula, in which we assume that so this is also known as the Hopper's formula. So, in this case we assume that all these 4 D's will be lost out in sawing, and the volume of our log that remains will be equal to G by 4 as the edge. So, it will be G by 4 into G by 4 into the length of the log. So, next we saw that it is around 78.54% of the actual volume of the cylinder. Now, it approximates the volume of timber that is obtained after sawing. So, that is one utility of it. Then we also saw some computations of how to use the quarter girth formula. Next, we looked up at volume computations in the field. So, one method is summing up the volumes of different trees that we can use. The next is the mean form height method. So, mean form height is given by sum of the volumes divided by sum of the basal areas of the trees, and we can also use pooled mean form height by taking measurements from a number of sample plots. And then we did this question in which we used both the form height and the pool form height to get the volume density. Then we also looked at the regression method of computation of the volumes. So, in this case we take the basal area and the volume, and then we use it to calculate a regression equation, and that regression equation is then used to calculate the volume density of the stand. Next we looked at volume table. So, volume table is a table that shows volume of a given species for one or multiple dimensions. So, those dimensions can be diameter at breast height, dBH and height, dBH height and taper. So, taper can be given in the form of form factor and so on. So, there are a number of tables that are included in the volume table. Now, depending on the number of variables, we have three kinds of volume tables. So, we can have a single variable, that is the dBH. We can have two variables, that is dBH and height for instance, or we can have multiple variables such as dBH, height, the form factor and so on. Then depending on the scope of application, we have general volume table, regional volume table and local volume table. We looked at their differences. Then depending on the outturn, that is what we are trying to measure, we can have standard round timber volume table, commercial round timber volume table and so on. Then we looked at the preparation of general volume table and local volume table by graphical method and regression equation method. So, in the graphical method we select trees, then we fell those trees, we measure those trees, plot the curves, do some checks and balances, and then use those to get the general volume table. And in the regression equation method, we define some generalized regression equations, and then we use the values of the trees that have been taken from the field to find out these regression constants, and from that regression constant we can then find out your general volume equation and this general volume table. Now, in the case of preparation of local volume table, we plot our GVT values of volume, diameter and height, and then we take our readings from the field, and then we draw a smooth curve to get the local volume table. Then we looked at its utility and limitations. We also looked at forest sampling, and we also saw difference between census and sampling. So, in the case of census, all the individuals in your population or all the units in the population are going to be measured, whereas in the case of sampling we just take a small sample from the population, take those measurements and then generalize them over the population. So, we looked at what is population, sampling unit, sampling frame, sample, sampling intensity and the kinds of plots. So, you can have plots of different shapes and sizes. Then we also looked at kinds of sampling. So, in the case of simple random sampling, all the individuals in the population have an equal probability of being a part of the sample. In the case of systematic sampling, we take every nth unit in the sample. In the case of stratified sampling, we divide our population into a number of strata and then we take samples from each strata. In the case of multi-stage sampling, we define large units as large samples and then we take subsamples from there. And in the case of PPS sampling, that is similar to the point sampling, the probability of a unit getting into the sample is dependent on its size. Next we looked at the density and mass measurements. So, the basic equations are density is equal to mass by volume. So, why do we need this density measurement? Because measuring the mass of large logs is difficult, but measuring their volumes is very easy. So, if we know the density of logs, then we can calculate the mass of the logs, and mass of the logs can also be used to calculate the amount of sequestered carbon that is there in the logs or in the trees. We looked at Archimedes principle. So, we get our sample using an increment borer. Now, mass is very much dependent upon the amount of water content. So, we can take these measurements and then we can find out the moisture content in a sample of wood. To measure the volume, we can either use these equations of volume like length into breadth into height if we have this cuboidal sample, or pi r square into h if we have a cylindrical sample, and so on. Or else we can go for a water displacement method, in which our sample is pushed inside a liquid with a needle and its mass is measured. Or we can use xylometer, that is this device to measure the volume of wood. Or we can directly measure density by using the buoyancy laws and by balancing the forces. Next, we also had a look at NDVI, which stands for the Normalized Difference Vegetation Index. So, NDVI is a technique that measures the reflectance of different surfaces to get information about those surfaces. So, we looked at true color composites, the vegetation spectrum. So, in the case of vegetation spectrum, we can utilize this red edge to find out vegetation and to differentiate it from different surfaces. Then we defined NDVI as NIR minus red divided by NIR plus red. So, in this two color composite, if we wanted to know whether this green color was because of vegetation or because of green colored tops of buildings that have been painted green, we can use an FCC. So, in the case of an FCC, we will display infrared in the red band, then red in the green band and green in the blue band to get these images. Or we can use NDVI. So, in this case, anything that is black does not have any vegetation, and as it goes towards whitish values, so it has vegetation. Now, this index can be used to measure changes in plant phenology. It can be used for vegetation and land use classification, biomass production, impact of grazing, maybe even impact of fire, amount of moisture in the soil, carbon sequestration potential of an area, and so on. So, for instance, we can use it to differentiate different surfaces, differentiate between different kinds of vegetation, discerning the health of a plant, and so on. Lastly, we also looked at site quality. So, in the case of site quality, we use this equation, area plus environment gives us the type and quality of vegetation. So, there are a number of factors that affect production of an area. So, for instance, rock, soil, climate, topography, vegetation, and so on. And site quality measures the relative production capacity. So, productivity is given as quality plus management input. So, if you keep all these management inputs as constant, that is fertilizer, site treatment, irrigation, grazing, soil compaction, growing stock manipulation, and other management inputs. So, if they are constant, then this equation of site productivity is equal to site quality plus management input will give us that site quality is directly proportional to the site productivity. Now, we can measure site quality using two methods. One is the CVP index and the second is vegetative characteristics. So, in the case of CVP index, we define this index I is equal to TV by TA multiplied by P multiplied by G by 12 and multiplied by E by 100. So, that will give us a value, and if that value is greater than 25, then we can have forest growth. Then we also looked at its limitations. Now, in the case of vegetative characteristics, we can look at the species that are there on a site, and we can also look at the characteristics of the tree in terms of DBH, basal area, height, volume, and so on. So, for instance, some species such as Palash are an indicator of degraded forest. So, if those species are there, then the site quality is not high. And if we have two forest sites that are having the same species, then if on one site our trees have larger volumes, then that site is having a greater quality. So, we saw how these factors can affect the growth of trees, and how in a standard way we use diameter and height curves to find out the site quality. So, in the case of site quality, we can use crop height method. So, in the case of crop height method, we get the top height and compare it with the yield table. And in the case of sample plot method, we plot diameter versus height curves and compare them with standard curves. So, like for instance, in this case, you have your site quality curves for 4 standard site qualities 1, 2, 3 and 4 plotted here, and for your site of interest, if you plot diameter versus height curve, so any curve that it comes closest to gives you the site quality. So, that is all for revision. All the best for your exams. Thank you for your attention, and Jai Hind. Do good.